Do you found the skeleton? How would you tell it's a reptile? You personally, how would you decide?
Well, interesting question. I don't know. I'd have to do some research on that.
Hey there YouTube, the Dapper Dinosaur here. We finally made it to the last part of this series in which Robert Carter disproves evolution by getting the science wrong. Which is ironic, since he actually has a biology degree. Once again, I'm addressing him directly since I know he's seen at least some of my stuff, and since he doxed someone else while trying to dox me. Plus, when he admitted I was right about something, then he couldn't be bothered to say my name. We just left off with Bobby telling us that people who don't even know who Gregor Mendel was were somehow influenced by him.
50 years later, they came up with this idea called neo-Darwinism. They invented mutations. Mutations, that answers.
They invented mutations? Are you saying mutations are fake? Because mutations are seen to happen in real time in the real world. Even if the concept was invented, it describes a real measurable phenomenon.
This whole thing, why does this fruit fly have red eyes and this one have white eyes? A mutation happened. All that Mendel stuff was just mutations.
What? No, he wasn't dealing primarily with mutations, but with pre-existing alleles. And the mutations still follow the basic laws of Mendelian genetics after they occur. Mutations didn't replace Mendel, they supplemented his work.
That worked until the 1950s.
What happened in the 50s? Did people decide that mutations didn't happen all along?
When mathematicians started analyzing evolutionary theory with math, they realized, you know what?
We're not trained in this field, but even if we were, we'd label our y-axis unlike John Sanford in the on-screen graph.
Even with millions of years, we can't explain the difference between man and monkeys.
Ooh, are we going to be getting into the waiting time problem? That'll be fun. But I won't jump the gun on you there, Bobby.
That's not enough time. And they dealt with that for a while. They invented junk DNA in the 70s, and that lasted until we sequenced the human genome. That's dead.
And then found that, yes, there is junk DNA. And it's the majority of the DNA. And by junk, I basically mean non-sequence constrained DNA, not useless DNA. Many of you have a junk drawer at home. Not because the stuff in there is useless, but because it's useful to keep around, even if much of it has little specific purpose and doesn't matter all that much most days.
But this is what's happened recently. Back in the day, way back in the day, Darwinism reigned supreme.
And people label the axes on their graphs.
Darwinists could explain, or at least claim to explain, all the science that we knew.
Bullshit. Never mind the fact that biologists don't try to explain science outside of biology, and ignore the fact that no one has been a Darwinist for nearly a century. But there has never been a time when there have not been open fields of research in biology since the origin of the field. If there's an open question in a field, it means it's a question that currently can't be answered.
But it's gotten away from them. The modern technological revolution has unveiled so many things inside life that Darwinism cannot explain, that their power of explanation has collapsed.
Ooh, what are these many things? Are you sure there are many things and not, nothing? Not that modern evolutionary biology has it all figured out, but the heavy implication is that there are things that preclude it, not just that are currently open questions.
I said this earlier, I'll say it again. If life were really simple, Darwinism might explain it.
No, that would be a problem, because since evolution proceeds only by modifying what came before, and often has to suffice with jury-rigging previous forms, it would predict that life in some cases is needlessly complex, like the mammal blood clotting cascade, or plant photosynthesis. Of course, that doesn't mean all life has to get more and more complex, but it would predict that some would, to the point of absurdity. Does the amoeba need a genome 100 times larger than a human? Probably not, but that's the kind of thing evolution would predict should happen in some organisms. Further, we've not been given any reason why complexity per se is a problem for evolution. If a complex trait is advantageous, why can't it be selected for? What would prevent a trait from increasing in complexity on occasion for mutation? Seems to me like there's no reason for either.
See, in Darwin's day, life, was based on a cell, but a cell was just a bag of salts. They called it protoplasm. They didn't know what was inside the cell.
They knew it was more than salt. And why are we talking about what Darwin knew? Darwin was ignorant of much, and wrong about much. Who cares? He's still not Pope of Science, nor was he ever. You know how much of evolutionary biology you can learn without ever once reading any of Darwin's works? All of it. It's all of it.
And if it was square or round, whatever, grew a spike or turned purple, I could imagine all sorts of changes in a really simple system. The problem is, the more we understand about complexity, the power of Darwinism to explain it goes the opposite direction.
Why? Don't just assert it, explain what the problem is. Otherwise, this is just the same as me saying the longer the sun keeps spinning on its axis, the less true Christianity becomes. Sure, I can say it, but it's stupid unless I can connect a reason the sun should stop to the truth value of Christianity.
And life as we know it today is not at all simple.
True, so because everyone agrees, I'm going to skip all your examples of it being complicated. I'm not here to sit around a fire and sing kumbaya about life being cool or complex. When you get to a point worth discussing, we'll pick this back up.
Any single particular letter in the genome is used, on average, in 6 different messages.
Yeah, I'm going to press X to doubt that each codon in the genome affects on average 6 different traits. But let's pretend you said a non-stupid thing and that you meant gene, because yes, most genes actually do affect more than one trait.
So that letter might affect your ability to digest garlic, how well you see in the dark, how tall you are, and what your skin color is. Not true, but it could affect 6 random things that have nothing to do with one another. Change that one letter, what happens now? Which trait is natural selection going to try to act on?
It doesn't have to act on one in particular, it acts at the whole organism level. So if the combination of those traits is beneficial, it will favor the mutation. If it's detrimental, it will act against the spread of that mutation. And if it's neutral, it won't act at all, and the mutation will be solely subject to drift. So in this example, if one of those traits, say night vision, is very useful, but the effects to garlic digestion, skin color, and height are minor in terms of reproductive success, then the mutation is beneficial. There's a thing called addition and subtraction, where you can just sum up the effects of change to multiple things at a time. Now granted, that's a conceptual thing. It's basically impossible to actually measure the effect of each individual change in most cases, but it's still the idea. The sum effect of all those fitness changes is what's going to be acted on by natural selection. This really isn't a high-level concept.
It can't. The system grinds to a halt if one change affects multiple different things that have nothing to do with one another.
Why? If there's a selected for trait, such as better night vision, that also happens to mean that a minor food source like garlic is now useless, but that's outweighed by the benefit of better night vision, then you get a population that abandons garlic and gets better night vision. That's just a trade-off. Evolution is already all about trade-offs. A bacterium will trade off the efficiency of a transmembrane transport protein to resist an antibiotic. A fish will trade off a higher infection rate for the ability to see. Humans might trade off the overall production of most digestive enzymes in order to produce lactase late in life to gain access to a new calorie-dense food. That trade-offs occur and that they can be complicated in no way means that natural selection grinds to a halt.
And life is so complicated that we absolutely know that that is true.
Life is so complicated that we absolutely know that it's true that natural selection grinds to a halt for no reason. Okay, buddy, that makes total sense and doesn't at all make you look like a raving madman who might be eating flies to collect their life essence for his master, who's on his way to England on a ghost ship. I'm going to recommend against your request for a kitten. And if you're in my audience and you got that, then maybe we can be friends.
You started out as a little cell in your mama, and then you split, and then you split, and then you split. Well, the little machines are copying the DNA. There's 3 billion letters in your DNA. Every time they make a copy, they make mistakes.
Yep, mutations.
You have 100 trillion cells in your adult bodies. You're right now carrying every single possible mutation. Essentially, every day, your body develops every single cancer.
Not really, but I get where you're going, and it's fine as an approximation.
Most of them are fixed, which is why you're still alive, but every once in a while, one escapes the fixing systems. It is estimated that a 50-year-old person has 60,000 mutations in every skin cell that they weren't born with.
Estimated by whom? When? Where?
Yeah, I'm going to be 49 this year. I'm looking at 60,000 mutations, and I've got millions upon millions of skin cells. Every one of them has different mutations. You are going to die of mutation accumulation if you don't get hit by a bus.
Or die of heart disease, liver disease, an infection, or some other calamity. But yeah, if you live long enough without any fatal injuries or other fatal diseases, cancer is sort of an inevitability.
We can't stop it. There's nothing you can do to stop. Well, you can slow it down maybe, antioxidants or don't smoke and things like that. Those things help. But in general, it's going to catch up with everyone in this room, and we're going to die eventually because your body is actually falling apart. That's genetic entropy.
Seriously, you were doing so well. Genetic entropy is a population phenomenon, not a phenomenon of individuals. If you want to make the analogy that a human is a population of human cells, then far from genetic entropy, cancer is runaway increase in fitness. It's evolution in action. And sure, eventually the whole population will collapse, but that was always going to happen anyway. While it exists, the cancer is by far the most evolutionarily successful group of cells in there, and they even evolve inside individual patients to be better able to avoid treatment and the immune system.
But it's not just falling apart on the individual level, it's also falling apart on the species level.
Not as far as anyone has ever been able to observe.
Because when we have children, we pass a small amount of mutations to our children. Every child born on average has about 100 mutations their parents weren't born with.
Sure, sounds about right. I don't know the actual pedigree mutation rate off the top of my head, but I won't contest this one.
Now, most mutations don't do anything.
Hey, what is this, like the third or fourth thing you've said that's actually flat out correct? Nice job.
They don't affect reproduction. They don't affect intelligence. There's 1 out of 3 billion letters. Change one letter. What's the chance that that letter is going to kill you? Very small.
Yeah, it's also a very small chance that it will do literally anything else. Most mutations occur in non-sequence constrained regions because that's a big majority of the genome. And of those that don't, a significant fraction are synonymous mutations anyway, which means it again causes no changes.
But it's like rust on a car again.
Is it though? Because that verifiably does something. That, in a calculable, measurable way, lowers the structural strength of parts. How does a mutation in a non-sequence constrained region do that? Oh, right. It doesn't. It literally, unlike rust, causes no change.
Our species, our genomes are rusting out over time. Natural selection is not improving us, it's just preventing us from going extinct quicker. That means actually it doesn't help Darwinism at all.
If only genetic entropy were real and not, mathematically impossible. I don't say this very often, right? In science, generally speaking, you can't prove anything. But genetic entropy is a fundamentally mathematical concept. It is trivially easy to prove, without any doubt at all, that genetic entropy isn't real. It's as fake as a four-sided triangle.
You ever heard, creationism is not testable. You ever heard that?
Yeah, parts of it are, parts of it aren't. It's too broad to simply say it's not testable, although I get why people say it. The important parts, like that maybe miracles happened, aren't really testable. But still, I try not to say it myself. I like a bit more nuance in my statements.
Evolution is testable. You're creation. You're just making stuff up.
I'm not sure how seriously to take that, but evolution is testable, and creationists do just make stuff up. But so does everyone. The important bit is, does the stuff you make up lead to testable predictions, and if so, when we test them, are they confirmed? If yes to both, then hooray! The stuff you made up is supported by evidence. If not, then boo, time to try again.
Well, that's not true. The Bible directly predicts one male and one female ancestor of all people.
Specifically that the human effective and census size was literally 2 at some point in the recent past.
Guess what's true in genetics?
That coalescence analysis can be run on the autosomal portion of the human genome, and it shows that they never had a population size as small as 2.
There's only one male and one female ancestor of all people on Earth.
Literally not the conclusion of any scientific study. If I'm wrong, Bobby, then just produce the paper that concludes that.
The evolutionists called her mitochondrial Eve and him Y chromosome Adam. They should have called him Y chromosome Noah, but okay, so they don't understand the Bible as well. They actually gave them biblical names anyway.
Who again is one common ancestor of all humans, not the first male ever, as even you know, but also not even the most recent common ancestor for all humans. He's just the most recent male human who can trace his lineage through all extant male humans via an all-male line.
The Bible predicts, because we only come from 2 people, you can't put a lot of genetic diversity into 2 people.
Fair enough, at most 4 allele per genome in the autosomal DNA, 3 per gene on the X chromosome, and 1 per gene on the Y chromosome. Not the Y has a lot of genes.
I can explain all the people in this room. You give me 6,000 years of mutation, and God front-loading Adam and Eve with some genetic diversity that's not mutation, and we can put all the people here into Adam and Eve.
No, we really can't.
We don't need millions of years. We don't need common ancestry with chimpanzees.
I'm glad you came up with a post hoc rationalization to dismiss the genetic evidence that clearly indicates that the human species is hundreds of thousands of years old, and that they are certainly related to chimpanzees. But guess what? Post hoc accommodations are basically meaningless. Evolution predicted the genetic evidence and got it right. Further, you can only get 2 humans 6,000 years ago by ignoring the autosomal portions of the genome, and then pretending that the pedigree mutation rates are the same as the phylogenetic replacement rates, which they're not. To get a pedigree mutation rate, you just check some DNA from a parent and their child, count the differences, and boom, mutation rate. But here's the problem. Some of the mutations in the parent and the child won't be in either's germline, so those are obviously not going to be carried forward to future generations. then only some of the mutations actually in the germline are going to end up in a new human, since most humans don't have enough kids that all of their mutations are going to end up in the next generation. Maybe if you're Genghis Khan who had like 1,000 kids, that's reasonable, but most of us aren't Genghis Khan. Further, even after mutations make it into a new child, there is selection and drift that weed out further mutations. All of this lowers the rate at which mutations accumulate in a population. And the thing is, this isn't something we have to guess at. There are locations like the Canary Islands and Tristan da Cunha that were settled not at some remote point in the past, but for the first time by humans in historical times, when people could simply write about the settlement, and the archaeological evidence corroborates these timeframes. Archaeology that young Earth creationists generally accept and that fits well within their timeframe. When we check these populations, we find that yes, their phylogenetic replacement rate is indeed slower than the pedigree mutation rate. And when we use that measurement to look for Y chromosome Adam and mitochondrial Eve, we get individuals who themselves almost certainly were not contemporaries, and who both also lived hundreds of thousands of years ago, not only a few thousand years ago.
The Bible also predicts one dispersal of humanity across the world. That's called the Tower of Babel in the Bible. The evolution is called out of Africa.
Those aren't synonyms. Those are competing hypotheses. In Out of Africa, Homo sapiens begins in Africa, probably around modern Ethiopia, and then spreads both into the rest of Africa and out into the rest of the world via the Sinai Peninsula. In the Tower of Babel idea, humans start in the Fertile Crescent and spread from there. These ideas make opposing predictions.
Why do they start in Africa? Multiple reasons.
Humans are more closely related genetically and morphologically to the African great apes than the orangutan, which is why the idea was first proposed. When the first non-human hominids, aside from Neanderthal, were found, it was primarily in Africa, with only later members of genus Homo spreading outside, including the aforementioned Neanderthals. The oldest H. sapiens fossils are in Africa, and the icing on the cake is that genetic diversity is highest in East Africa, linkage disequilibrium is lowest there, and all non-African genetic diversity is a subset of one portion of African genetic diversity. It's pretty conclusive. Why, Dr. Carter? What do you think the reasons are?
I think there's some latent racism here, because Darwin and his disciples always believed the Africans were closer to monkeys.
Well, no matter what they thought, Africans, along with the rest of humanity, are so close to monkeys that they just are monkeys. You don't get to escape your ancestry. All humans, regardless of where they're from, regardless of what they look like, regardless of any past or present oppression, are all equally monkeys. But regardless of that particular belief, which I neither care to verify nor refute that Darwin and his friends held, the reasoning and evidence remains conclusive. Humans came from Africa.
And therefore, of course, we evolved in Africa.
No, that wouldn't be sufficient. That would allow for humans evolving in Africa, Asia, and South America at least. And for a while, the multi-regional hypothesis was actually popular and was far more racist. It was the idea that humans independently evolved from monkeys and apes in several places around the world, and so a Chinese human, an African human, and a South American human might actually be separate but related species, essentially.
Today, there's a lot more diversity in Africa than other places in the world. And there's two ways to do that. Either you have an older population, or your population went through more generations.
Well, there's no particular evidence to think that Africans in prehistory had significantly shorter generation times than other humans, but it still doesn't help with the fact that non-Africans are a subset of Africans. They nest within the African groups. Everyone outside of Africa is more closely related to some Africans than those Africans are to other Africans. You have to explain that, and just pointing out a rescue device for the higher diversity in Africa isn't going to help with that.
There's lots of different ways to get differences between populations, and they're basing the entire thing on the fact that some Africans have more mutations than the rest of the world put together.
And that the earliest fossils of humans were found in Africa, and that the rest of the world isn't just lower in diversity, that it's nested within the African diversity. That's a problem you can't just hand wave away by saying, I don't know, Africans just have babies young and they mutate a lot.
But how do I know that's older? What if they're just like more mutated than everyone else alive today?
Then they wouldn't be recovered in every rooted phylogenetic analysis as encompassing all other diversity. They'd be a sister group to either all non-African diversity or a subset of non-African diversity.
If that's the case, then the origin of people isn't the place where everyone's spread out from.
The origin of people isn't the place that everyone set out from. I don't even know what you're trying to get across there.
Think about it. The out of Africa theory says that small people groups traveling into uninhabited territory migrated with one Y chromosome and one mitochondrial ancestor in the recent past, even for an evolutionist, through the Middle East.
Basically, yeah, there was one exit to the Middle East that eventually gave rise to the rest of non-African humanity.
All of those are things you would directly predict from the Tower of Babel account in Genesis.
No, you'd predict that humans would expand from the Middle East and that some subset of them would go into Africa, while others went into Europe and Asia, and from Asia into Australia, Oceania, and the Americas. Which would mean that African humans should be a sister group to non-Africans or nested within some group of otherwise non-Africans. That's not what we see.
But they don't want you to think that way.
You're right, I don't want people to think in a way that's directly counter to the evidence.
I explained that, by the way, in this talk, Mitochondrial Eve and the Three Daughters of Noah.
Might be something for me to look into.
All right, I'm going to hit this subject very briefly. The origin of life. You see, if evolution can't explain the origin of life, then evolutionists need to go home.
You see, if sculptors can't explain the origin of granite, they need to go home. The fact that a particular explanatory mechanism has a starting point that isn't basically just the beginning of all things doesn't invalidate it. Evolution remains true whether or not science can explain the origin of life. In fact, even if life were miraculously created, that doesn't falsify evolution. Evolutionary biology isn't here to disprove God or form an explanation for all phenomena. It's about the diversity and change in reproducing biological populations over time. If you don't have any such populations, then you don't have biological evolution.
Darwin tried to avoid it. He joked. He said, you might as well talk about the origin of matter. Yeah, it's called Big Bang, Darwin. They talk about it now.
Yeah, because now cosmology has testable predictions about such things.
Because people realize if we can't explain the origin of matter, what are we going to have? God explain it?
Maybe, it probably wouldn't upset the theist who also doesn't reject evolution. And while it wouldn't be a scientific explanation, it might still be the right one. But we probably shouldn't stop looking into scientific ideas just because, like in all things, someone has said, yeah, but what if it was a god?
You can't invoke God. That's not naturalism. What if science was just a way of thinking that might help to explain things? So one way of thinking is Zeus is standing on top of a mountaintop, and he's got his lightning bolt ready, and when I get out my petri dish and I do an experiment, he's going to either zap my lightning bolt or not zap my lightning bolt. That is a way of thinking. It's not really very conducive to science, is it?
Hence why it's not a conclusion that science can reach. But scientists sure can.
You've seen this before, I'm sure. Come on now, there you go. Boom, you've seen this, right? The famous Miller-Urey experiment. They've got gases bubbling up, and they're zapping them with electricity, and they're dripping down here. Do you know what they're making down here? You know what that is? It's just random molecules.
Among which are specifically amino acids, which until then had not been shown to form spontaneously in abiotic conditions. It was thereby proof of the concept that amino acids could predate life. That's all the experiment meant to do. The question was, can this class of very important chemicals that life needs form without life? The answer was yes. Since then, scientists have gone on to do better experiments in conditions more closely approximating that of prebiotic Earth and have had even more success. But I'm betting you aren't going to talk about any of that there, Dr. Carter.
Because random chemical reactions produce random molecules. You are not made of random molecules. You're made of a very specific set of molecules.
Some of which were made in that experiment. And it's not like abiogenesis, that is the beginning of life from non-living chemicals, involved the spontaneous formation of humans or any other modern living thing.
There's this many potential molecules that are made in that reaction, and you're made out of this many.
I fail to see the relevance. No one has ever thought that the Miller-Urey experiment demonstrated how life came about. It wasn't meant to. It was meant, like most experiments, to help answer one specific question. Again, that question was, can amino acids form in abiotic conditions? Again, the answer was yes.
That is not supposed to be true, but it is.
In science, nothing is supposed to be true. That's the kind of thinking that leads to a conclusion first, evidence later thinking. The kind of thing creationists like to do.
Worse, there are molecules that you absolutely depend upon for life that can never be made in this reaction.
Yep, that's true, and it's still irrelevant to anything.
They're too fragile. As soon as they touch one of these tar molecules, they're destroyed instantly.
Since the tar is where the samples were collected in the experiment, then, if that were true, wouldn't the experiment have failed to show that it created amino acids? Are you now accusing Miller and Urey of fraud, Dr. Carter? Because it's either that, or you're lying, or you're massively incompetent. Again, I ask you to let me know for future reference.
The origin of life, they cannot explain most complex organic molecules. Yeah, you can make some, fine.
Amino acids, RNA, lipid bilayer membranes, and even amino acid polymers can all be formed in labs in prebiotically relevant conditions.
But not DNA.
DNA almost certainly was not present in the first life, so there's no particular reason to think it needs to be explained as some abiotic process. We know RNA chains can form abiotically, and that's all that's needed for the genomes of the first organisms. In fact, RNA chains can auto-catalyze their own formation, and evolve without being in a cell. Further, they can catalyze the formation of proteins all on their own with no ribosome needed. The fact we can't spontaneously create a modern cell doesn't mean anything for the possibility of life to form without a miracle on a prebiotic Earth.
Not proteins.
Right, but proteins can be formed by RNA, which itself can form spontaneously.
They can't make polymers.
RNA is a polymer. It forms spontaneously.
This is funny. You are polymers. You have long chains of DNA, long chain proteins, and long chain sugars. Long chain fats. That is you. This is what you're built of. Long molecules. All life, from the simplest bacterium to astronauts, require long molecules. The problem is, the polymerization reaction that makes those molecules, a chemist would never do it in the presence of water.
Weird, because that's where it happens in life. In water.
Because water always breaks the bonds apart, and it breaks it down so fast that chemists get rid of all the water because it'll destroy their proteins, their fats, anything they're trying to polymerize.
You may or may not be aware of this, but all life lives in and around water. Water is the solvent of organic chemistry. And while it's true that water will hydrolyze various organic polymers, it does so at a particular measurable rate, and if the rate of synthesis is higher, then the system will contain a persistent level of these organic polymers. That's all it takes. If the system is making polymers just a bit faster than the water degrades them, then they get to stick around on net.
So life had to start with no water?
Nope, that's just a straw man.
Worse, the last one there. How do you get instructions out of random molecules?
Any genetic sequence of any given length will have a non-zero proportion of amino acid sequences it codes for that have some further biological activity or function, assuming it's long enough in the first place. Based on experiment, that fraction seems like it could be as high as 10% for sequences of about 100 base pairs, and if we allow for a single mutation, even higher, perhaps as high as 60%. So basically all that has to happen is the process samples a portion of sequence space about half as big as the functional portion of that space, and we get a 50-50 chance of making a functional instruction, if you will. Of course, more experiments are needed, but the fact is random sequences can and do have function.
See, DNA is not just a molecule. It's not just a code. It's a set of information. Essentially, you have an encyclopedia inside every one of your cells that tells, you start with a single cell, and the instruction set in that cell said, make you.
Cool, but what with synonymous codons and the predominance of non-sequence constrained regions, there are lots of possibilities for a genome that would make a phenotype exactly identical to your own.
Without that instruction set, you can't have life. But that instruction set would never arise from random mistakes. Random mistakes produces genetic entropy. It destroys life.
Well, since as we've already covered, random sequences can and do have function, and genetic entropy simply isn't a thing, there's not much more I need to say.
You couldn't start with a bunch of disorganized letters and randomly say, okay, let's just say you got a Scrabble set, right? And you pull out enough letters to spell out a chocolate chip cookie recipe. Is that ever going to happen?
No, human language is a poor analogy for DNA. It's a great analogy for evolution, though.
No, and if it did, someone accuse you of cheating.
And reasonably so. For English, the fraction of functional alphabetic sequences of any given length is far smaller than what it seems to be for DNA, which is why it's a poor analogy.
There is nothing in the origin of life in chemistry that suggests it's possible.
I don't know. The more than 5 million results on Google Scholar for, quote, origin of life, unquote, might disagree. As with the fact the field of abiogenesis is still putting out new research and making progress. Just in 2022, at about the end of the third quarter, there were almost 63,000 papers related to it that had been published. For example, in February, Tanya Bose et al. Reported methods for protein synthesis without a ribosome. Is it all figured out? No, and it probably never will be, but to pretend that the Miller-Urey experiment is the height of origin of life research and then just pretend that there are no mechanisms for the formation of molecules like RNA or proteins is just wrong.
In physics, that suggests it's possible. In probability, no way. Information theory?
Gotta love it when a creationist talks about information. Just like with kinds, it never gets to a useful definition that we could use to make predictions that could be tested to see if the creationists are right.
These are massive branches of science. All of them argue against the origin of life from natural processes.
Funny how the experts in those fields disagree. But I'm sure a marine biologist is going to set all the physicists straight.
Including this little guy. You know what that is? That's the world's smallest rotary engine. And just like our motors, it's powered by electricity. But instead of electrons, this guy is powered by protons. Hydrogen nuclei, H+.
It's the bacterial flagellum, isn't it?
Those protons make this thing spin. There's a rotor, there's a stator. Oh, and it takes this thing called ADP, that's phosphates. That doesn't exist in nature. Phosphate's too reactive.
Oh man, if only Yukio Yagamata hadn't reported abiotic synthesis of ATP and ADP back in 1999 in the paper Prebiotic Formation of ADP and ATP from AMP, Calcium Phosphates and Cyanate in Aqueous Solutions. Now, is his reported mechanism the answer we need for the formation of these chemicals? No, but it's at least proof of concept that these can form in nature. Plus, why is this relevant? No one is suggesting that the bacterial flagellum was around in the first life. It came about much later than that, probably hundreds of millions of years after ATP started being made by cells.
And it bonds it to this thing there called, this molecule is so reactive, you never find it in a free state in nature.
Probably why no one is suggesting that this particular protein complex was original to life.
This little thing here spins in circles and makes those purple things flying off. You make your own weight of this molecule called ATP every day.
Okay.
But you never find ATP in nature.
That's not what the literature says.
Why did life decide to use ATP? And how did it find it before it started?
Dunno, and it found it possibly in the wild, or it used something else like breaking hydrogen sulfide bonds, and only later evolved to use ATP.
Now I'm going to give you another really interesting thing that happens inside your cells right now. There's your piece of DNA again. DNA is a very long, fragile molecule. Now we're going to see something zip by here. This is a polymerase. Woo, there it goes. We're doing a little magic here. We're not showing all the complex things of getting things inside it. They're just floating in there magically. This polymerase is going by. It's unzipping the DNA. It's adding nucleotides to it. It's making a copy of the DNA. It's making a copy of a gene. Now, your genes are much more complicated than this. Your genes are spliced and diced before they're ready to be used. We're just really oversimplifying it just to show you what's happening. Now, when it gets to the end of the message, it says, stop. Message kicks out. But what's that thing good for?
For being copied into another strand of RNA, which then goes to a ribosome to be translated into a protein.
That RNA molecule has to be translated into protein. And we use these amazing little things. These are called transfer RNAs. At the bottom end of each transfer RNA, there's this 3-letter code that matches 3 letters in the DNA. And they go into this machine. It's called a ribosome.
At the center of which is a ribozyme, which just happens to be RNA, which again can in fact synthesize protein on its own. Ribosomes are far better at it, but they're also the product of evolution. The first life wouldn't have been as efficient as modern ribosomes.
And inside the ribosome, which also has error checking mechanisms in it, it takes these messenger RNAs, lines them up, literally pops an amino acid onto the growing protein strand. That's happening inside every one of your cells millions of times every second, right now. But most proteins, if left to themselves, they'll fold into a blob and be not useful. And so these other molecules, they're called chaperones. They come and they grab onto the protein strand to prevent it from folding in properly. And they bring it to another molecule. We don't quite know how this thing works, but we know what it's shaped like and we know what it does. It's called a chaperonin, that little watermelon-shaped thing there. They stick this unfolded protein into the chaperonin. It then does this little magic and spits out a 3-dimensional folded protein.
Which is indeed very cool, but I'm not sure how that, a part of modern life, is relevant to abiogenesis. The 3 kinds of chaperonin in life are themselves evolved structures. You can't point to any one of them and then extrapolate that back to the first life. And would that mean that the first life had relatively unreliable protein synthesis? Yes. And would selection pressure favor more reliable protein synthesis? Almost certainly. And would the first life have to immediately compete with highly efficient protein synthesis? No.
We started with the 1-dimensional letters A, T, G, and C and translated it into a 3-dimensional with 20 different letters of all the different amino acids in a protein. By the way, this depends upon ATP.
It does now, but bare ribosomes don't need that. But you probably don't know about that, since apparently you think that all research into this topic stopped 70 years ago.
That might be part of the ATP synthase engine. This is a chicken and egg problem now.
It was a chicken and egg problem, but like the literal chicken and egg problem, it's been solved. Eggs came first, and ribozymes came before ATP.
Because all life uses ATP, all life needs the ATP synthase. But you need ATP synthase to make ATP. You need ATP to translate the proteins that are part of the ATP synthase. Actually, it's a three-way chicken and egg problem.
Okay, Bobby, I want you to every day for the next year look into a mirror and say to yourself 10 times, the first life was not identical to any modern life.
If you don't have any of those, you're dead. How did life start?
Much simpler and less efficient.
And why on earth would it be so complicated?
A combination of selection for more reliable or efficient mechanisms, coupled with the fact that evolution can't start over. It can only jury-rig what it has already.
And why? Literally, it's translating from Swahili to Sanskrit to Old Norse to computer programming language.
I see you've forgotten what literally means, because I can guarantee you that DNA isn't in Swahili and RNA isn't in Old Norse, nor is it in computer code. As we all know, it's actually in Arabic, al-lugha al-arabiyya.
Why would it do that?
Because rather than being designed by some super smart dude, it's the result of happenstance, chance, and selection by a mindless process rather than a mind.
Unless it was not evolved by itself.
Yeah, because all designers remember to make sure that their instructions have to be translated like 4 times before the end product can be made. That's just how you do it. And hey, that's your analogy. I think it's a bad one, but it's not really working here for you.
All right, I'm running a little long. I'm going to try to hurry up here. My last section, science and morality. Before I go there, I want to show you one more big idea. Think of the smallest thing that you can see.
That's an object about 1/10 of a millimeter in size, so larger grains of dust. Got it.
Think of the shortest time that you can notice, a blink.
That's harder to figure out, since the brain actually tends to filter out blinks. Humans have a variable flicker fusion rate, but by around 50 Hz, humans generally can't tell the difference between flicker and smooth motion or solid light. So the smallest amount of time a human can notice is probably on the order of 10 milliseconds or so. Interestingly, blinks happen on the scale of tenths of a second, so far longer than the smallest time a human can notice.
There's a limit to what you can directly observe. Think of the largest thing you can see in the longest time you can experience your lifetime.
The largest thing you can see is probably the Andromeda Galaxy, which is just barely visible with the naked eye on a very dark night.
There is a zone of science that we can absolutely know, see, feel, touch, taste, whatever, right here. Now we can build things like microscopes and telescopes, and we can expand our zone of direct observation.
Thanks for the 4th grade science lesson.
But evolution is not here.
Tell it to all the evolution experiments going on in labs right now that use tools and direct observation to watch evolution happen in real time.
Evolution is out here.
Some of it is, but also that time axis isn't nearly long enough. The effects of deep past events can be directly measured, and predictions about new data regarding them can be made. Evolutionary history is based in part on those direct measurements, and predictions are made about future data from the deep past, and like we've already gone over, it is frequently confirmed. Creationism doesn't do this. Where it can make non-trivial predictions that are falsifiable, they are all either already falsified or so far untested, and curiously, the creationists seem slow to bother to test their ideas. I wonder why.
Evolution is in the zone of deductions only. You can't directly test it.
Sure you can, the same way you directly test for paternity.
You can't directly see it. It is a historical science that knows nothing of the laboratory.
Wait, so the Lenski lab that is running the Lenski long-term evolution experiment, that's not a laboratory? What does the lab part mean then?
Ouch. Bill Provine, he was a colleague of Dr. John Sanford's at Cornell, committed evolutionist. He said, "Let me summarize my views on what modern evolutionary biology tells us loud and clear. There are no gods."
I don't care about this guy's metaphysics, so I'm going to skip them. This is no more relevant than me cherry-picking a Christian who thinks that science leads them to God and who does believe in God in the afterlife.
You realize that if you believe in God, the evolutionist believes that you're carrying a mutation that you inherited from your monkey and fish ancestors that causes you to believe in God?
No, it's more that your genetics certainly enables you to have a brain complex enough for such concepts, and there may be a genetic component to a propensity to believe certain things, but I don't think anyone falls so heavily on the side of nature that rather than nurture as to say that your belief in God is determined directly by your genetics. How would that account for people who change their belief in God, either gaining or losing it, or both during their lifetimes? Or how would it account for people who grew up in non-religious households who become religious, or vice versa?
You realize that the hardcore evolutionists think you're just a machine? Every thought and reaction of your mind is just chemicals. And the reason those chemicals exist and do those things is because they helped you have babies 100,000 years ago.
Hey look, an argument from consequences. It's fallacious. No matter what unpalatable results evolution might lead to, they don't make it wrong. Giving reasons you don't want evolution to be true won't change that it is. So until you stop and make arguments against evolution per se, I'm skipping.
But I wanted to impress upon you how profound the subject is.
There, I can agree. The question of where humans came from is one of profound importance for their understanding of themselves as biological beings.
How empty Darwinism is, both in the science and in the philosophy.
You probably should have done better than basically getting all the science wrong then.
And how salient in today's world the Bible still is.
Not to science it's not, because science doesn't get to start with a conclusion. If your interpretation of the Bible were right about this, then we wouldn't need to presuppose it to get to the conclusion that it's right. The evidence would simply fall in line with that. It doesn't.
It's not an empty book.
Well, yeah, it's full of all sorts of stuff.
It's not a book without power. It's not a book without explanation. Actually, it does a great job. Now, it's not a science book. It's a history book.
Not all of it is even that, and the parts that are were written before history as we know it was even conceived of. So the so-called historical narratives in the Bible were written for polemical reasons, not as sober objective recountings of events.
But as a history book, it shouldn't be wrong about the science when it talks about science.
Well then, that bit about animals looking at stripy sticks while mating and then having stripy babies might be something you want to address.
And when we look at all these different things, all these vast fields of science actually fit into biblical history. That is amazing. I didn't know that when I was growing up.
It sure would be amazing. Got any actual evidence that isn't, wrong to back that up for the parts of science it is at odds with in your mind?
And I hope some of you now know it better than you did. And that's the first time you've ever heard it. I hope I challenged you.
Well, there was some challenge. I had to look up about 12 sources to write this whole 40-page script.
I remember being challenged when I was 18. I was like, this is crazy. This can't possibly be true. And now I believe it. But now I've got a lot more information than I did back then.
Well, a lot more misinformation.
So it's for you. Please take this and use it as you can. And take it and use it to impact somebody else's life.
I'll do my best. And after this, it's just some prayer. I have nothing to say about prayer, so I won't. Well, that was a long video full of a lot of nonsense and apparently outright lies. Dr. Carter, if you're listening, I'm open to you telling me why it is that all this demonstrably false information was given to your audience. I suspect, though, that if this video series gets any response from you, it'll be in the form of a whiny blog post with no attempts to address any of it, or any real attempt to fix your own errors. As for my audience, I hope you enjoyed this far more than I enjoyed writing the script. If you did, why not like the video and leave a comment? If you didn't, tell me why not in the comments, and dislike the video. Either way, if you want to see more of my content, be sure to subscribe and turn on all notifications so you're always notified when I have more content. I'm the Dapper Dinosaur.
Thanks for watching, but before you go, I just want to say a special thank you to my channel members and my patrons, especially those pledging $20 or above. Bob Knob, Work in Progress, Benthoven, Denny5252, Ian Chen, Landon Knoll, Mabity Babbity, McSpooks, Sphincter of Doom, The Venerable Bede, and Patrick Dennis. As you probably know, YouTube is a very volatile platform, and from month to month my income on the channel can swing wildly. But the people you see on screen are directly supporting so that I don't have to worry too much about that, and the channel can keep going as it is, and perhaps even improving. If you'd like to join this team, there are links below to join the channel or in the description to join the Patreon. The Patreon starts as low as $1 a month, and the channel memberships start as low as $1.99 a month. On Patreon, you can even get a discount for pledging annually. If you do decide to pledge, you'll get access to an exclusive Discord server just for channel supporters, as well as early access to all of my pre-recorded videos, often up to 2 months in advance. Higher tiers will unlock even better perks. Now, if an annual or monthly pledge isn't right for you, but you still want to support the channel, there is a merch store down in the description, as well as an Amazon wish list, just for me. And if financial support isn't something you can or want to do, then if you still want to help out the channel, please just like and share these videos and make sure you comment on them. It really helps the channel out. Thanks again for watching.